Worldwide, there are currently more than 130 million people suffering from diabetes. Some of these people can only survive with regular injections of insulin. In 1982, recombinant insulin was first used. This was the first protein medicine produced on an industrial scale using genetic engineering techniques. Pancreas cells first produce one protein chain called proinsulin. Then the middle section, C-peptide, is cut off from it. In this way, two protein chains are formed, alpha and beta, combined into one molecule of biologically active hormone. During the production of recombinant insulin, the constructed vectors separately introduce DNA-coding beta chains and DNA-coding alpha chains into Escherichia coli cells. After mixing the isolated and cleaned protein products of these genes, the active hormone is produced.
Next, insulin. What is insulin? Insulin is a polypeptide hormone produced by beta cells of the pancreatic islets of Langerhans. We know our pancreas found a cell islets of Langerhans. This islets of Langerhans cell contain four cells: alpha cell, beta cell, alpha, beta, gamma, and PP cell. Alpha cell is generally secreted glucagon hormone. Beta cell generally secrete insulin hormone. Delta cell generally secrete somatostatin hormone. PP cell generally secrete pancreatic polypeptides. So, insulin is generally secreted by beta cells of islets of Langerhans of pancreas. But when any abnormalities for the beta cell damage, for the insulin tolerance, and many reasons, when beta cell facing a more pressure and also adverse condition, day by day beta cell is death or also the dysfunctioning. Then it works slowly, and the main function of insulin, it plays a central role in regulating blood glucose level. It also a profound effect another metabolism of protein and lipids, and displays some mitogenic activity. Some of these mitogenic effects are likely mediated by the insulin-like growth factor 1 receptor, and their physiological relevance is questionable. So, the main function of insulin is for the decrease of glucose level in our blood system, in our circulatory system. Just insulin metabolize glucose to produce energy that's need. And when a large number of insulin found in our circulatory system, insulin transfer the high level of glucose in cell. Therefore, glucagon utilize the required amount of glucose for the growth, development of different metabolic processes. When the high amount of glucose level in cell, glucagon also transferred the excess glucose in blood. So, the opposite function perform insulin and glucagon. Insulin maintain the glucose level in our circulatory system, and glucagon maintain our glucose level in cell. When the insulin activity is lower, then glucose level is increased in our circulatory system, basically blood, then create an abnormal symptoms called diabetics. Then we use insulin hormone as a vaccine to maintain the blood glucose level. So see the structure. We know insulin generally consists by three chains: chain A, chain B, and chain C. Chain A, chain B, and chain C. Chain A generally consists, both chain consist by amino acid. Chain A consists, chain A totally both chain A, B, C, those chain generally consist by amino acid. When the pre-insulin is converted to mature insulin, chain C is removed, then only two chain exist, chain A and chain B, and those chain are generally attached by disulfide bond. See the S and S, S1 and S2, on and 2 mono, therefore 2 di. This bond is called disulfide bond. So insulin chain is generally consist by disulfide bond. Chain A and chain B, when found, it is mature insulin. A question generally I given your previous semester final, why we do not administer insulin orally. আমরা কেন ইনসুলিন মুখে খাই না? Why insulin inject by blood vessel? See the structure, a easy answer. Insulin is generally consist by two chain, chain A and B, and both chains are generally attached by two disulfide bond. Disulfide bond. When we intake it orally, the insulin is generally digested by our digestive juice and break down the disulfide bond. Then the two chain, chain A and chain B, is separated, and it generally exists as a dysfunctioning level. Then it do not work properly against the metabolism of glucose level. For this reason, we do not consume or intake insulin orally. Generally, it is administered by injection directly to blood vessel. Properties of insulin: onset, peaking time, duration, concentration, route of administration. First, onset. Onset is the length of the before insulin reaches the bloodstream and begins the lowering blood glucose level. Peaking time. Peak time is the time during which insulin is a maximum strength in terms of lowering blood glucose. Duration, how insulin continued to lower blood glucose. Concentration, also the properties and also the route of administration. We administered insulin by injection of the blood vessel, not orally. Important functions of insulin: it stimulates the uptake of glucose by muscle and adipose tissue, it stimulates glycolysis, it stimulates glycogenesis, it stimulates protein synthesis, inhibits gluconeogenesis, inhibits lipolysis. Those are the major functions of insulin. Why insulin is not administered orally, already discussed this point. Next, production of recombinant insulin. How can we product recombinant insulin? By genetic engineering technique. See the outline. First, we select a bacterium containing plasmid DNA, and we also select a human cell that contain gene, that contain first DNA, and we know DNA contain a specific gene. Therefore, we cut vector by restriction enzyme, and we also cut the DNA as a sticky end. Therefore, we ligate vector DNA with a specific DNA by ligase enzyme. Next, it produce, and this DNA is called recombinant DNA or genetically modified DNA or hybrid DNA. Then this DNA contain one side a vector and another side is a specific DNA. Then the DNA will transfer the DNA to a specific host and also a model host. Generally, we use a model host of organism, E. coli bacteria, E. coli. Plasmid is reintroduced into bacterium. Therefore, engineered bacteria multiply producing insulin. We use the specific DNA that contain insulin producing gene, and we collect the gene from the pancreas islets of Langerhans, beta cell of islets of Langerhans of pancreas. Then engineered bacteria multiply producing insulin. Therefore, engineered insulin is separated and purified to produce human insulin. And vialing it for inject the diabetic patient. It is the main procedure for the production of recombinant insulin in E. coli. Then we use the recombinant insulin to the control of glucose level in the diabetic patients. It is the outline. So you read in this section, define insulin structure, just we see for your learn. Then properties of insulin, function of insulin, therefore why insulin is not administered orally. Next, production of insulin, just outline.